Hey guys, today we're going to be discussing the process of gene expression and protein synthesis. Transcription begins in the nucleus with initiation, where the RNA polymerase enzymes collide the DNA at the initiation site. Within the RNA polymerase, the DNA gets unzipped, allowing them to begin reading and recording the information within the DNA. The information takes form of nitrogen bases, adenine, thymine, guanine, and cytosine. As the polymerase reads these bases, it takes nucleotides within the nucleus and pairs the bases with the following nucleotides: cytosine with guanine and adenine with uracil, vice versa. The pairing is referred to as complementary pairing, with uracil is a base found within the RNA that takes place of the thymine in the DNA. As the RNA polymerase moves 5 to 3, which is referring to the nitrogen-based carbon layout within the 5 at the top and the 3 at the bottom, it creates a long elongated chain of nucleotides. RNA polymerase isn't perfect, however, it can cause mutations within the nucleotide chain. Such mutations can be substitution, where the wrong nucleotide is added, insertion, where the RNA polymerase adds another nucleotide extending the chain, and deletion, where it just forgets about one altogether and leaves it out. Some of these have no effect at all, but others appear drastic. Transcription ends with termination, where the RNA polymerase reaches the termination site of the DNA and releases the nucleotide chain as pre-mRNA. Once transcription is complete, the next step, of course, is splicing. Splicing is the editing of pre-mRNA by which spliceosomes or SNRPs attach themselves to the pre-mRNA, cut out non-expressed information. This is done by the pre-mRNA being divided into sections called exons and introns, by which the exons are the expressed and are needed sections for future protein synthesis, while the introns are not and can be pinched right off and recycled. The process of pinching is where the SNRPs pinch off the intron and then bring the exon ends and connect them. Once all is left in one RNA chain for the only exons in the chain is now matured mRNA, or mRNA in general, which stands for messenger RNA. Now that the editing is complete, we must move on to translation, where the mRNA has to move outside the nucleus and the cytoplasm and the RER on the hunt for ribosomes. Translation. For the mRNA to be used for processing into proteins, the mRNA must make contact with the smaller subunit of ribosomes, followed by the larger one just after. This ribosome that attaches to it is also known as rRNA and does not create proteins but polypeptides. Once the ribosome attaches to the strand of the RNA, it is able to process the RNA in the required gene going in the direction 5 to 3 again. The 5 to 3 direction being yet again the 5 representing the phosphate group at one end and the 3 being the carbohydroxyl group at the other. Once the ribosome reaches the codon, first codon is AUG, which stands for start, the process can begin. tRNA carrying amino acids are constantly floating around in the cytoplasm. Rapidly in large quantity, these components make contact the mRNA in the catalysis site through entering the A site for access. tRNA sees for its anticodons, or amino acid base, will pair up with the mRNA's codons through complementary pairing, like transcription. The tRNA's amino acid connects in the catalytic site, the ribosome known as the P site, elongating the polypeptide chain. Once the connection is made, a new tRNA comes in and repeats the process. This new tRNA coming in and moving in the previous one happens to the E site, where they are ejected. Once the anticodon matches up with UAA, UAG, and UGA, the polypeptide is released, as this is the stop codon. The final protein can be used within or outside the cell for stuff ranging from cell structure to enzymes. This is the end of the protein synthesis. Thank you for listening.